Hi, my name is Barbara McPherson, and today we're going to discuss blood constituents and their functions. Blood is classified as a fluid connective tissue that consists of components or formed elements. Blood is a fluid that transports oxygen and nutrients to the cells and carries away carbon dioxide and other waste products. Technically, blood is a transport liquid pumped by the heart to all parts of the body, after which it is returned to the heart to repeat the whole process. Blood is considered both a tissue and a fluid. It is a tissue because it is a collection of similar specialized cells that serve a particular function. These cells are suspended in a liquid matrix, the plasma, which is what makes the blood a fluid. Blood makes up 8% of the whole of your total body weight, and you typically have somewhere around 5 liters of blood in the body. Blood is an opaque red fluid. It freely flows throughout the body, but it's denser and more viscous than water. The characteristic color of blood is imparted by hemoglobin, which is a unique iron-containing protein. Hemoglobin brightens in color when it is saturated with oxygen, and it's termed oxyhemoglobin. And then the blood color darkens when oxygen is removed, which is termed deoxyhemoglobin. This is the reason why when you cut yourself, and if it's like from an artery, the blood is bright red. If it's from a vein, it's a darker colored red, because the veins in the artery are oxygenated. The blood in the artery is oxygenated. The blood in the veins is deoxygenated, so it has a different color. Okay, the components of blood consist of formed elements that are known as cellular elements. They consist of erythrocytes, or red blood cells, leukocytes, or white blood cells, and platelets, and plasma, which is extracellular matrix. Blood cells are formed in bone marrow, and blood and each of the formed elements have a specific function. We'll discuss in more detail the function of blood the next slide, but just to go over it right here, it provides the body with oxygen and removes carbon dioxide, transports nutrients and hormones, regulates body temperature, and brings waste products to kidney and liver for removal. And here's a picture of the components of blood in a test tube. And it also shows you under each one kind of what they do. In this one, it says plasma transports nutrients, hormones, and proteins. It's a yellow liquid that makes up about 55% of the blood's body volume. We'll get to more of that later. Platelets form clots to stop bleeding. Platelet makes up less than 1% of the blood. Red blood cells carry fresh oxygen through the body and remove carbon dioxide. Red blood cells make up about 40 to 45% of the blood. White blood cells is part of the body's immune system. It detects and fights viruses and bacteria. There's 5 major types of white blood cells, and they make up less than 1% of the body. Okay, the function of blood. Blood has several functions, but the first function of blood is that it carries oxygen to body cells, and it removes carbon dioxide. So that's kind of showing you the picture there on the left is kind of showing you blood moving through there carrying oxygen. The second function is to carry nutrients to the body and deliver hormones. The picture on the right is showing you that, where it's nutrient absorption from this is the small intestines and the villi and the capillaries from the small intestine where the blood picks up the nutrients from. The third function is to help regulate body temperature. And the picture on the left shows the blood vessels, how they dilate and how they constrict, and how heat loss works with each one. Um, the fourth function is to carry waste products to the kidney and the liver for removal. The picture on the right shows a picture of the kidneys and how it will go into the renal artery, the renal vein, and then out the ureter for waste. That is the function of blood. Okay, the function of the formed elements, we touched on that a few slides back, but red blood cells, the function of red blood cells, which is the picture on the left, transports oxygen. The picture on the right shows white blood cells, and their function is to fight, again, to the body's immune system. This picture is just kind of showing you what they look like. The red blood cells are disc-shaped, they're flexible, and they're bioconcave, which means they've got a little dip in the middle. The picture on the right, again, is showing you the types of white blood cells, and we'll get to that in a minute when we get to the composition of blood. Functions of the formed elements. The main function is for the platelets is clot formation. So what happens when you cut yourself, you start to bleed, the blood will start to clot up. These platelets will rush to that site, they will clot up, and then you'll have some proteins called fibrin that'll come in there and mesh it all around and kind of make a little, like a little net that will kind of stop the bleeding. We're not going to go into a whole lot of detail of the function of these formed elements, but that's basically what the function of platelets are. Platelets are little bitty small fragments. They're not a complete cell. They're just little fragments of the red blood cell. Okay, now the function of plasma. It has several functions, and here's a picture showing you plasma, which is the very top part of the test tube that's kind of yellow in color. So plasma helps maintain blood pressure and volume. It supplies critical proteins for blood clotting and immunity. It carries electrolytes such as sodium and potassium to the muscles. It helps maintain a proper pH balance in our body, which supports cell functions. Okay, now we'll get to the composition of blood. Okay, so when you take a blood sample and you put the blood in a centrifuge, which is a machine, you set the tube in there and it spins it real fast. And what it does is it separates the components of blood so that you are left with the plasma on top, the formed elements on the bottom, and a little layer in between those two that's called a Buffy coat. So, the centrifuge will separate the blood components, and then the denser, heavier material will go to the bottom, and the lighter material will remain on the top. So the composition of blood is as follows. So plasma is 55%. The formed elements are 45%, and then the Buffy coat here is 1%, actually less than 1%. Okay, now the elements that make up these components, the plasma consists of about 91% water, 7% protein, and about 2% other solids. So if you look over here to the right side of the picture here, underneath proteins, it'll tell you how many percent the proteins have and what they are. So there's albumins, globulins, fibrinogens, and prothrombins. And albumins is 57%, globulins is 38%, fibrinogen is 4%, and prothrombin is 1%. So that's the breakdown of proteins inside the plasma. The other solutes that's in there, the 2% of other solutes are ions, nutrients, waste products, gases, and regulatory substances. Those all make up the 55% of volume that the plasma has. Remembering that most of plasma is water. Okay, the 1% here, the less than 1%, is the Buffy coat. And if we, the Buffy coat is where you find your leukocytes, your white blood cells. Even though it has it here in formed elements, it's actually in the Buffy coat. Okay, and then your white blood cells consist of, or your leukocytes consist of neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And all of these help the body fight um, infection. Each one of them, oh, I'm sorry, it went to a different, um, went to a different slide. Sorry. Each one of those, though, will help the body do different things. And then I know I'm not on that slide anymore, but if you look down to the formed elements where the red blood cells are, the erythrocytes, um, I can't tell now because I'm not there, but I can't tell what they were. But the remaining 45% of the blood is made up of red blood cells. And there's only one type of red blood cells, and that's just red blood cells. So now we're going to go over here and we're going to discuss stem cell research. Red blood cells, white blood cells, and platelet producing cells are all descendants from a common precursor. They are a hematopoietic stem cell. Now, stem cells divide asymmetrically, which means one daughter cell remains a stem cell of the same type, while another daughter cell acquires a new identity. For hematopoietic stem cells, which are found in the bone marrow, one daughter cell remains a hemopoietic stem cell, while the other goes off and differentiates into some other different kind of stem cell, either a myeloid stem cell or a lymphoid stem cell. Myeloid stem cells and lymphoid stem cells also divide asymmetrically, with their non-stem cell daughters generating the mature cell types of the blood. Myeloid stem cells give rise to red blood cells, platelets, and other types of white blood cells. But lymphoid stem cells give rise to the types of white blood cells classified as lymphocytes. And so here you can see in the picture here that it all starts from one blood cell up here at the very top, the hematopoietic stem cell right here. And then it's going to differentiate into either a myeloid stem cell or a lymphoid stem cell. And then the myeloid stem cell is going to break down into to all of the white blood cells with the exception of the lymphocyte. So it's going to break down into what is called a megakaryoblast, a proerythroblast, a myoblast, and a monoblast. The megakaryoblast will form a megakaryocyte, which is what forms your platelets, as you can see if you follow the arrow down. The proerythroblast will form a reticulocyte, which will form your erythrocyte, which is your red blood cell. The myeloblast here, if you follow that down, it's going to form 3 of your white blood cells, your basophil, neutrophil, and eosinophil. And then the monoblast, if you follow that down, that's going to develop into the monocyte. Okay. But if you go over here to the lymphoid stem cell, that's going to differentiate into a lymphoblast. Now, the lymphoblast is in turn going to differentiate into a natural killer cell, which is a large granular lymphocyte, as you can see in the picture, or a small lymphocyte, which the small lymphocyte are going to differentiate into 2 different kinds of cells, a T lymphocyte and a B lymphocyte. And both of those lymphocytes are big in fighting infection. Okay, so this is just a precursor to what we're going to talk about in the research that we're talking about here. So in the research, The research is over induced pluripotent stem cells, which are called iPSCs. They're derived from somatic cells through a reprogramming process, which converts them to a pluripotent state. That is a state of embryonic stem cells. So basically what it's trying to do is take embryonic stem cells. It's trying to take these pluripotent stem cells and get the same kind of reaction that you can get from embryonic stem cells. So over the past decade, iPSC models have found interesting applications in the study of human diseases, with blood disorders featuring prominently. Since the emergence of induced pluripotent stem cells, stem cell technology, which is a little over a decade ago, iPSC models have held the potential to bridge these two worlds: primary patient cells and conventional disease models. It does this by direct relevance to human disease. So being patient-driven while enabling precise, controlled, and scalable experiments, it was only possible using traditional model systems. So here in this picture, we review the use of iPSC model, in the study of blood diseases, mainly encompassing two areas: the modeling of gene and cell therapy of inherited monogenic disorders, and the modeling of myeloid malignancies. Effectively, all disease modeling applications involve differentiation of the iPSC lines of interest, along with their control to the cell types in which the disease manifests itself. So, mutations associated with blood diseases rarely cause molecular or phenotypic changes at the pluripotent state. So, the appropriate hemopoietic cell type needs to be generated and studied by a process of directed vitro differentiation. This is where the iPSC are introduced to exit the pluripotent state and commit to a mesoderm fate. So it's kind of showing you this breakdown of all of this, of how it's working. Here's the mesoderm state. They're doing all of this like in a lab, of course. They're growing all of this and then taking it and then producing these cells over here. So it's followed by an induction of hemogenic endothelium, a specialized form of endothelium that gives rise to blood during development, and subsequently by the specification of this hematopoietic lineage. So that's kind of showing you what this that's what this picture is showing you, that they're taking these mesoderm cells, this human cell here, and then they're transforming them into these type cells. Okay. The next one, or the next picture, is the same study, and it's shown where the application of iPSC models of human diseases fall under two categories: investigation of disease mechanisms towards classic drug development, and development of gene and cell therapy strategies. So this one says, in the first, iPSC lines serve as research reagents toward therapeutic discoveries. The second, they serve as therapeutic agents themselves. So the study focused on the two areas of disease modeling for blood disorders: the preclinical modeling of gene therapy of inherited blood diseases, and the modeling of blood cancer. So this iPS-derived blood transfusion products, it's not a model that just like beats all other models, but it fits the purpose for this particular given application. So that's why they use this type of model to see if they could get these differentiated cells, get these stem cells from an isolation of a somatic cell. So, and what makes this so good, what makes these iPSCs good, what makes them good is that you can study their molecular mechanism of a specific disease-causing mutation. They're good for studying the mutations, but they're not the best for studying complex diseases that you get in adulthood or older age. They're better at at getting it when they're young, when they're younger. So it's better for those, and it has limitations. Basically, is what I'm trying to say. But it's a best model to fit what they're trying to do with stem cell research. Um, with there being such a issue with stem cell research due to the fact that the best stem cell that you can get is embryonic stem cell, but there's so much issue and controversy surrounding that the best way to come up with that without all that controversy is try something that this research is doing, this iPSC model, to try to take somatic cells, program them to pluripotency to make stem cells so that you can fight blood disorders and blood cancers and other types of diseases. Okay, I hope you've enjoyed it and I hope you learned something. Thank you very much and have a nice day.